everyone, I'd like to go over muscle contraction, specifically the excitation-contraction coupling that takes place. This is the process by which we get the message from a nerve to a sarcomere. The nerve tells the muscle to contract, but the contractile unit is that sarcomere, if you remember from previous videos. So the sarcomere needs to get the signal. So how do we get it from the nerve to the sarcomere? Alright, so it starts with an action potential coming down the axon. If you remember from chapter 4, each nerve has an axon, and this axon is what transmits the signal from the cell body of the nerve. And so that signal is called an action potential. It has a potential to cause an action. So action potential. An action potential is like a message that's traveling down the axon of the nerve. And it gets to the end of the axon, which is the axon terminal. If you think of like airport terminal, that's the end of the hallway, is the terminal there. So an axon terminal is in the end of the axon. And this is where the action potential is headed. Now, when the action potential is coming down the axon, it's depolarizing the membrane. Remember, we talked a little bit about different channels and depolarization of the membrane. Well, what happens is it comes across these calcium channels right here. I drew them in green on either side, and those calcium channels respond to the depolarization in the membrane of the axon. So as that potential is coming down, they feel it and they say, ooh, ooh. And what happens is calcium is going to go into the cell. Calcium comes in. And when calcium comes in, we have synaptic vesicles. They're really just vesicles with proteins in them. And that says, hey, now's the time, release what you have. So these vesicles come and, get the right color. So right now they're up here. We have these vesicles holding proteins, and they want to release them, but they don't know when. So when this action potential comes down, it stimulates the calcium to come in. Calcium says, hey, now's the time. And these vesicles come, and they fuse with the membrane. So here it is fused. And the contents then leave the vesicle. So the contents, the proteins there, have now left the vesicle. And they're in this open space, which we call the synaptic cleft. Wherever a neuron, wherever an axon terminal ends and talks to something else. So here's my axon terminal. Here's my other cell. Wherever it's talking to something else, that's a synapse. So we have a synaptic cleft, the opening, the space between the axon terminal and whatever it's talking to. Synaptic cleft. And in this case, it's a muscle cell. All right, so these little proteins, which have a name, they're acetylcholine. Acetylcholine. They diffuse from the axon terminal to the muscle cell. Now they're going to a special place on the muscle cell. So they're crossing the synaptic cleft. They're going to something called the motor end plate. And the motor end plate is a special, look how I drew it. I drew it wavy. So instead of being nice and flat, it's wavy. Its membrane is bunched up. And why do we have that in the body when we have, remember which line is longer? If I had two pieces of string, which one would be longer? The wavy one, right? We have more surface area. So this surface area, the increased surface area, allows for more events to take place. So let's talk about what events need to happen on this motor end plate. The spot on the muscle that the axon terminal is right up near. It's not touching. We have that synaptic cleft. But on this, we have special channels. Remember we talked about how there are voltage-gated channels and ligand-gated channels? Well, here are the ligand-gated channels. And we have something called a ligand, what's going to bind to that channel, and that's that protein that's being released. So we have acetylcholine being released. It's going to bind to our ligand-gated channel. So here it is binding. Here it is binding over here. Binding right here, binding right here. So let's pause and back up. We have our axon, action potentials coming down, triggers calcium to come in. Calcium comes in and tells it to release those vesicles. So it releases the vesicles, and the proteins inside, which are acetylcholine, go out into the synaptic cleft. And they diffuse across the synaptic cleft, just diffusion. And they get to ligand-gated channels. And they say, wow, we fit here. We're the right ligand. And so acetylcholine binds to these sodium ligand-gated channels. This is really important to know that they're ligand-gated. And it's important to know that acetylcholine is the ligand. So acetylcholine binds to these sodium ligand-gated channels, and they open. So then what happens? Sodium goes in. And when sodium goes in, what happens? Is sodium negative or positive? Sodium's positive, right? But what's the membrane inside the cell? It's negative, right? So we have this influx of positive ions. And what's that going to do to the membrane potential? It's going to flip it, right? So it's going to, instead of being negative, it's going to flip it. It's going to be positive. And if we have enough of that happening, we can get a membrane depolarization all the way up away from the motor end plate, and we can trigger another type of channel. What are those? I drew them in light blue here because they're still sodium channels, but now they are voltage gated. And when it's voltage gated, does anything need to bind to it? We don't need anything to bind to it. What are these voltage-gated channels responding to? They're responding to a change in the membrane polarity. So it's really responding to a depolarization in the membrane. And we got that over here because the acetylcholine was released. Acetylcholine bound to the ligand-gated channels, and it opened them. And we had a depolarization here at the motor end plate. And it was strong enough that we stimulated our voltage-gated channel. So now, this voltage-gated channel has been stimulated. And it depolarized. And so, let's see. So sodium is going to flow in. Now when sodium flows in, that's going to depolarize the membrane. So the next one's going to be stimulated as well. And the next one. And it's kind of like if you've ever been to a baseball game or any sporting event where they do the wave. Have you ever seen that? So the person over here starts the wave, and the person next to them does it, and then the person next to them does it, and then the person next to them, and it goes all the way around the stadium. And so that's kind of what's happening here. This sodium channel depolarizes, and this sodium channel depolarizes, and then this one, and then this one, and then this one. And it's this huge ripple effect across the membrane of the muscle. So, but we also have another type of channel, don't we? Because we can't just leave it depolarized, right? So after it closes, what happens? We have potassium-gated channels. And then potassium is going to leave. And again, that's going to happen as a ripple effect. But the sodium, they're still depolarizing. So the signal's traveling down, down, down. And then, remember the T tubules? It travels down the T tubule. And if you remember, we had a triad by the T tubules, right? So we had the sarcoplasmic reticulum on either side, and a T tubule down the middle. So what happens is, as the membrane starts to depolarize here, it's going to trigger special channels that are in the sarcoplasmic reticulum. And these special channels are going to open in response to the membrane depolarization. These special channels are calcium channels. I drew them green again. I kept calcium green for the whole, across the board. So we've gotten the message from the neuron, right? And it started with these ligand-gated channels. But the depolarization triggered voltage-gated channels. And voltage-gated channels, this is the key. Because without these voltage-gated channels, it's not going to travel as a ripple effect. You would need acetylcholine binding all the way along here. And that would be really hard to do. But because we have voltage-gated, it just travels like a ripple effect. And we get down here, and it depolarizes the membrane in the T tubules, stimulates the sarcoplasmic reticulum, calcium channels. And you know what happens? Calcium leaves the sarcoplasmic reticulum. So instead of being inside of there, it's now outside. And what does calcium do? Why do we want calcium? Why do we care about that? Isn't calcium going to bind to the troponin over here? So we have our sarcomere, if you remember. We have actin and myosin, but we also have tropomyosin and troponin. Remember, tropomyosin is covering those actin active sites, and troponin is holding tropomyosin in place. But when troponin binds to calcium, it pulls the tropomyosin off the actin active sites. And actin can bind to myosin, and we get that contraction taking place. So let's review this. This is a lot. So we have depolarization when action potential coming down the axon gets to the axon terminal and stimulates calcium channels to release calcium. That tells the synaptic vesicles to fuse with the membrane and expel their contents, to release their contents into the synaptic cleft. They diffuse across the synaptic cleft and bind to sodium ligand-gated channels on the motor end plate. That causes a depolarization. And if we have enough acetylcholine released, it causes a big enough of a depolarization that it stimulates these sodium voltage-gated channels. Key that it's voltage. And that causes sodium to go into the cell. And then we get the wave. We have sodium released, potassium channels bring it back to normal. Sodium, potassium, sodium, potassium, sodium, potassium. So wherever there's a sodium channel, there you're going to have membrane depolarization. And then that's going to be followed by membrane repolarization by the potassium channels. But it's going to be a ripple effect because they're voltage-gated channels. They're responding to a change in the membrane potential. So we start here, depolarization, depolarization, depolarization is traveling across the membrane. Then we get to the T tubules. It goes down the T tubules, and it's going to, because if you remember, the T tubules are part of a triad. It has sarcoplasmic reticulum on either side. And as the membrane in the T-tubule starts to depolarize, it triggers these calcium-gated channels, these calcium channels, sorry, not calcium-gated, calcium channels in the sarcoplasmic reticulum. So these are, let me write this out here. Calcium, they're voltage gated. And I drew them in green right there. It might be hard to see. But I want you to know that in the sarcoplasmic reticulum of the triad, we have calcium voltage gated channels. And those channels respond to a depolarization as the action, or as the membrane potential depolarizes, traveling down the T-tubule. So what happens? Those calcium channels get stimulated. They release calcium from the sarcoplasmic reticulum. Because remember, sarcoplasmic reticulum, its main job here is to store calcium. So it stores the calcium, releases the calcium. Calcium binds troponin. Troponin changes shape, pulls the tropomyosin with it when it does. Tropomyosin then moves off of the actin active sites, and the myosin heads are there waiting, ready to grab onto those active sites, and they pull it over. Does that make sense? It's pretty involved. It's also really fun when you get it. So the whole goal here is to get from the axon, the message in the nerve, to the sarcomere, the contractile unit. And we cannot do that without voltage-gated channels. We have voltage-gated sodium channels that depolarize the membrane. We have voltage-gated potassium channels that repolarize. We have voltage-gated calcium channels that are in the sarcoplasmic reticulum and respond to a membrane depolarization in the T-tubule. All right. I'd say any questions, but it's a video. Email me if you have any questions. Let me know. And take care.